talk with you, more especially to all the students. I am very happy to see all the students behind the scene, and I would like to get more questions from the student side. So I'm talking today. Professor Ratnakar already described me all about. I think she described me more than what I know. So I need not to say anything to introduce myself. Today I'm talking about the Scarless corneal wound healing and gene therapy. Can you go to the next? This slide, actually I added this slide. I'm becoming more romantic and Sirena being in the Lucknow, watching this beautiful image of eye. And definitely the eyes are the most beautiful creation of the nature. And it's utmost important to maintain the beauty of eye by, can you go next? By maintaining more importantly to the cornea. Because cornea provides 2/3 of the reflection to the eye, and this is the second most outer layer of the eye. The first is the tear film, and it is immune privileged. It protects the eye and avascular and dome shape. And cornea has 3 main layers. Outermost is epithelium, innermost is endothelium, and in between is stroma. And in between there is the Bowman's membrane and Descemet membrane. These are the single cell layers, and this is the stroma makes about the 90% of the cornea. More important of the cornea feature is transparency. No matter what, the cornea should be transparent to maintain its function. If there is any injury or trauma happens to the cornea, it may cause blindness. So can you go next? So corneal blindness is one of the leading cause of eye blindness, and it is epidemic in Africa and Asia due to trachoma. And any injury or infection or surgical procedures like PRK may cause corneal blindness, and it has a huge economic burden worldwide. It's more than 70 billion burden of economic burden worldwide. Can you go next? What is the status in India? India has more than 6 million individuals bilateral cornea. Both eyes have the corneal blindness, and more than 25 to 30,000 ads every year. And if you see this figure, you can see almost entire India is suffering from the corneal blindness. So why this is so burdensome? Why this is so important to study? So at these few slides I made the mechanism behind why this corneal wound healing is so important. What is the problem? And these are few teaching kind of slides for the students, for the beginners. When any injury or trauma happens to the cornea, it breaks the outer layer of corneal epithelium, and that releases the battery of cytokines like IL-1, TNF-alpha, that activates the native cells of cornea, that is keratocyte, and then it proliferates and migrates to the site of injury, and that is known as fibroblast. And the cytokine that releases from the tear gland and epithelium, it also releases TGF-beta. The TGF-beta helps in the transdifferentiation of fibroblast to the myofibroblast. And then by physiological condition, if it is the injury is grade of injury is not too grave, it may go to normal condition. Apoptosis happens, it goes in normal condition, and transparency maintains. But if it is going in pathological condition, if there is no apoptosis, it causes age, and that myofibroblast release alpha, SMA, collagen, all the fibrotic proteins, extracellular matrix proteins that accumulates around the cells, and that makes the cornea age or corneal fibrosis. So this is the main function mechanism that drives the corneal fibrosis and cause the vision loss. So what are the current therapies available so far? So there are many therapies, chemotherapies, surgical interventions like anti-fibrosis, anti-VEGF, because neovascularization is another problem with the corneal fibrosis, and many steroids are available, and surgical intervention that has frequent but not free from post-surgical complications happens after the surgical intervention. So these, though there are the therapies available, they have severe side effects. So what is the next? Next is corneal transplantation. You all know the corneal transplantation is available and happens around, but still it has some limitations because there is long queue, long waiting list in the in what US like countries where more organ donation awareness, but India has a long queue of the organ donation and corneal transplantation. So corneal transplantation is also has certain limitations. So we cannot provide to everybody those who need corneal transplantation. So what next we can do? So next is again. So it has annually approximately 1.85 corneal transplants performed globally, and it needs a lot more cornea. So what next is gene therapy, that we offer gene-based therapy. So this slide is very general as far as students to understand what actually is the gene therapy. Gene therapy, that healthy gene of interest that we need to either edit or replace with the help of the faulty gene that we can pack in the viral vector or loaded with the nanoparticles delivered to the site of like cornea, your organ of interest, and it replaces or edit the faulty gene. So it has some benefits like long-term benefits, infrequent clinical visit, single administration, reduce patient compliance, and safe given locally. And gene therapy for the cornea is very easy, topical, just put a kind of eye drop, gene that is going to be used for therapy. So cornea is very ideal for gene therapy, but because by visual observation you can see the effect of the gene therapy. So this is what gene therapy that we want to address to mitigate or get rid of corneal fibrosis. These are certain genes in ocular gene therapy that is being used, is being tested in the different in vivo and in vitro studies. So what is our hypothesis? What we are doing in our research? So localized and controlled AAV mediated or nanoparticle based therapeutic genes delivery into stroma, because stroma is the main part where the fibrosis happens, because this fibroblast keratocyte is the main cell part of the stroma, the fibrosis, because epithelium heals after every 3 days because of the limbal stem cell. Eyes around there is limbal stem cells that replace the epithelium, the outermost layer of the cornea. It replaces every 72 hours because it is outer cell layer, and it gets more trauma with rubbing or dust particle or any infection, and it gets replaced every 3 days, and your epithelium is healed and you are okay. But if the injury is deep enough and goes to the stroma, then we have to deliver the gene to the stroma and define technical method, cornea neovascularization and scarring in neo without significant side effects. So we are we are having two kind of approaches. One is monotherapy, just targeting single gene that we can use, but there is another approach that we can combine two different combination gene therapy, where we combine multiple 2 or 3 genes to take multiple pathways at a time to get rid of this problem. So current gene therapy that is we are targeting HGF, BMP7, decorin, VEGF, and SMAD7. These are the few genes that we are testing in vivo and in vitro studies. Go next. Okay, so this is in vitro model. First of all, we have to have an in vitro model to test of our gene of interest for the gene therapy. So we use human cornea that we get it from that corneal transplantation, but somehow they are rejected. They are not fit for corneal transplantation. So they ship us to us and we use for the primary cell culture. So we get the primary cell fibroblast from this human cornea, and with the help of TGF-beta in serum-free media, after 72 hours we get myofibroblast. This is in vitro model of fibrosis that we get, and then once we get this, we can test whatever the gene of interest that you want to test as a gene therapy. So this is the model that we use for in vitro studies, and we are targeting TGF-beta pathway because TGF-beta follows SMAD pathway. That TGF-beta, SMAD2, and after phosphorylation, it activates, and SMAD2 and SMAD3, it makes a complex of 2, 3, and 4. Without 4 making this all complex, this will not enter into the nucleus. So we are targeting, because this is the one of the main root cause of trans-differentiation of fibroblast to myofibroblast. We are targeting this pathway to somehow to regulate or control to the formation of myofibroblast. So other than that, in this pathway there are several co-suppressor and co-activator in this TGF-beta SMAD pathway. There are certain listed co-suppressor and co-activator. If we activate co-suppressors and if we knock down co-activators, we can modulate the corneal fibrosis. So we have chosen certain genes, TGIF1, TGIF2, P300, and CVP based on the literature and other studies, previous studies. We tested. We activated the suppressor and knocked down the activators. So we tested the TGIF CRISPR based activation of TGIF1. You can see the result here is this green signal is the L5 that is hallmark of fibrosis, corneal fibrosis. If we see that TGF-beta treatment, it has robust L5 expression when there is no activation of TGIF. When TGIF was activated, you can see the more TGF, less L5 positive cells. So the next, and the activation of TGIF also suppress the pro-fibrotic genes. We tested certain fibronectin, collagen 1 and collagen 4, that if you see the CRISPR TGIF activation plasmid, when we use, the pro-fibrotic genes get down-regulated at mRNA level. So likewise, we tested the TGIF2 again. TGIF2, when the TGIF was activated, the L5 level was down. So another thing, that combination of, here I am giving the example of combination of suppressor and activators, both. One is activated, one is suppressed. So what is the effect? That some preliminary data, this is the very preliminary data, that's when the combination effect has more, is combination of activators and suppressor has more effect to suppress the pro-fibrotic genes and L5 markers. So another thing that we are testing is SMAD gene silencing. Here I described how this pathway happens, TGF-beta and TGF-beta follows SMAD pathway. So there is SMAD2, SMAD3 and SMAD4. So silencing of these three proteins, if it is SMAD2, SMAD3 and SMAD4, we can see the L5 or less L5 positive cells, we can say, when these three proteins were silenced. And there is SMAD7 also. SMAD7 suppress the formation of this complex SMAD2, and if we overexpress gain of function, if we do gain of function of the SMAD7, overexpression of SMAD7 suppress the formation of alpha SMAD positive cell. So this is another way how to tackle this pathway, TGF beta SMAD pathway, to modulate the myofibroblast formation. So next is in vivo model that we use once we get these genes in vitro stage. Then we test. The next is the animal model. We test the genes for gene therapy in the animal models. The best model that we are using is the rabbit. That is the New Zealand white rabbit. We use this rabbit eyes for the corneal wound healing and gene therapy. We create injury wounding with the help of because the anatomy of eye after the the anatomy of eye is very similar to human rabbit eye. So we do the wounding with the help of 0.5 normal NaOH, with the help of 8 mm soap with 8 mm filter paper. We keep it on the mid of cornea and let it go for 30 seconds, and then go for corneal examination. You can see just after injury, the injury, it has a lunar kind of effect. It goes because the epithelium is completely burned. So you can see after the injury, you can see here, and slit lamp examination shows this is very fine slit lamp that shows the epithelium is very healthy, and but in alkali burnt cornea, you can see the slit is narrow beam is become wide, so it means it is got reflected, and because of the burn or the epithelium, injured epithelium. This is the slit lamp examination that we do through the rabbit, and then we do the testing for the other clinical examination. This is phytometry that test the corneal thickness, because corneal thickness is also because just after injury it has very huge inflammation. In general, the rabbit cornea is around 360 to 390 micrometer, but after injury it become like 700 to 900 micrometer. This much inflammation happens to the so thickness is also another parameter to test the health of cornea and epithelium. And the intraocular pressure, obviously, IOP is another thing that we test, because any injury, though IOP varies with the circadian rhythm, but we test the intraocular pressure with the help of chromopen. If increased IOP may cause glaucoma. So glaucoma is another condition where lot more pressure, normal range is 8 to 11 mmHg, but if it goes up to 20, it causes glaucoma condition. It can break the retina, damage the retina. So next is, and then we test the confocal imaging for that gives the entire health of cornea from outside epithelium to the endothelium. Is give the whole picture like CT scan kind of thing, around 200 to 400 images at a time it gives, and we can analyze all the data. So we tested the combination of BMP7 and HGF gene therapy. Go next. In the rabbit, here this is the stereo microscope images. This is minus therapy, and after 21 days of injury, when the therapy was there, you can see it's almost like knife one. So after injury, and when the therapy was there, BMP7 and HGF, we can see the cornea almost transparent. But those have not been treated, you can see there is some haze that is corneal fibrosis still remains. So this combination therapy is really working very efficiently. Go next and see the slit lamp examination at 21 days minus therapy. The inflammation is still there because slit B beam is very thick, and here when the therapy, those having seen the therapy, the slit beam is become narrow and almost similar to the knife one. So next, and after the at 21 days, we euthanize the animal, we collect the cornea, and then we do sectioning, and we do all the molecular biological techniques to see the level of different profibrotic and fibrotic markers like alpha-asma and others. Here we can see the more alpha-asma positive cells in minus therapy one. Those having not seen the therapy, but those at therapy is 21 days, it reduced alpha-asma positive cells. That is also showing that the BMP7 reduces alpha-asma and reduces the corneal blindness. And similarly, we tested the other profibrotic genes, fibronectin, collagen, and you can see the therapy one has reduced, all the therapy group has reduced collagen 1, fibronectin, and alpha-asma at RNA level. Then, once you test your gene of interest for therapy, it is also very important, if it is safe to treat, you go for the next level for the human trial. So safety is also another concern. Is it safe or not? So we keep our animal for next 3 months to 6 months. Next project is we are keeping the animals for next 1 year even. Though it's very costly to maintain the animals, n is equal to 12 or 18. It's very costly because the per dime of rabbits is very high in US. And so, but we tested a 6 month post AV decorin, and this at 6 months you can see that they are safe. There is no side effect. The cornea is very transparent. There is no redness, no side effect was found even after 6 months. So it seems that the decorin, AV5 decorin gene therapy is safe for the in vivo studies. And this is the confocal images that showing the epithelium to endothelium. This is the epithelium cell layers, and the intermediate epithelium. This is anterior stroma, this is posterior stroma, and this is the endothelium. Endothelium has very hallmark feature, that hexagonal honey bee-like structure. If there is any change in this hexagonality, it means the endothelium is not good. Endothelium health is not good. And the cell number is also, cell density CD percentage is also very important, because it does not regenerate. Once it is degenerated, it widens. It's saved to fulfill all this space. Its shape become larger but does not grow another endothelium. So count of hexagonality is also very important to see the health of endothelium. Endothelium is important because endothelium, the layer, it pumps the water from cornea to aqueous, because hydration of cornea is also important. More than 5% hydrated cornea will cause opacity. Cornea should not be hydrated. So this words like sodium, potassium pump is there that pump out all the water from the stroma to the aqueous. So this is also very important to maintain the corneal transparency. So the summary is that CRISPR-mediated gene therapy activation has promoting physiological wound healing in injured cornea, and localized topical BMP7 GLP therapy is corneal fibrosis, and restore transparency in vivo with excessive healing and rendering selective apoptosis, myofibroblast, profibrotic SMAD silencing, antifibrotic SMAD7 overexpression, and its combination studies to control TGF-beta mediated fibrosis, and 6 months follow-up as it is safe. There is no side effect of gene therapy, and potential, and this recurrent gene therapy is potential for corneal fibrosis and neovascular disease. So gene therapy may offer hope for millions of people affected by corneal blindness. So yeah, go ahead. And I acknowledge my mentor, Dr. Rajiv Mahan, and all the lab members that we have. They have contributed a lot to get this work done. Without their support and help, this work cannot be done. So this is my journey. Professor Banerjee already introduced me, but this is Java University. I graduated from Nahar University, Masters from Jiva University, and PhD from Banaras University. I work for Eye Institute, and currently I am assistant professor at University of Columbia. Me and Professor Banerjee has the BHU connection as well. So this talk, I think, is taught with more collaborations, we have more collaborations, and make stronger connections. Thank you.